Welcome back, everyone. If you're a focused learner gearing up for those GCSE biology exams, you are in exactly the right place. Today, we're putting together the ultimate high-energy animation explainer of how your body actually processes what you eat. We're looking at the whole digestive system, going all the way from that sandwich on your plate to the actual fuel powering your cells. Okay, let's dive into this. So let's start with what sounds like an incredibly simple question, right? Why do we actually need to digest our food? It seems obvious, but framing it exactly this way is the absolute key to understanding the entire topic. You see, digestion is just an incredibly elegant evolutionary solution to a major biological problem. We eat huge things, apples, bowls of pasta, whatever, but the fundamental unit of our body is the microscopic cell. There is a massive physical mismatch between the giant food we consume and the tiny format our cells can actually use to stay alive. To get really specific, it basically comes down to basic chemistry. The carbohydrates, proteins, and lipids we eat are large, insoluble molecules. And because they're insoluble, they simply cannot be absorbed into our bloodstream. It's like trying to fit a sofa through a mouse hole. Our digestive system's single overriding goal is to break those massive chains down into small, soluble molecules. Only then can they slip right into the blood and be carried all around the body to the cells that desperately need them.
All right, section 1, the digestive organs, our body's biological assembly line.
So, how exactly does the body pull off this incredible disappearing act? Well, it uses a highly specialized biological assembly line. The journey kicks off right in the mouth with mechanical chewing, travels down the esophagus, and into the stomach where things get highly acidic. From there, it moves into the long coils of the small intestine, which is where the main absorption happens. Then onto the large intestine, where water is absorbed, and finally, out through the anus. So the crucial point is that the food follows this continuous tube. But notice the liver, gallbladder, and pancreas sitting just off to the side? Food never actually passes through these accessory organs, but as we're going to see, we absolutely could not digest our food without them. Moving on to section 2, digestive enzymes, our biological catalysts. Okay, so while chewing and churning break our food into smaller physical chunks, we really need chemical digestion to solve that insoluble molecule problem. Enter enzymes. Think of them as vital biological catalysts. They're essentially molecular scissors perfectly designed to speed up chemical reactions and snip the chemical bonds holding our food together. Because enzymes are large proteins, they fold into highly specific 3D shapes, and this brilliantly illustrates the whole concept. Notice the little indented area on an enzyme? That's called the active site. Only a specific molecule, which we call a substrate, will perfectly fit into that active site, exactly like a specific key fits into a lock. Once they bind, the reaction takes place, the bonds are broken, and the newly freed, smaller products are released. Now, for your exams, you absolutely need to know the big 3 enzymes rapidly. First up, carbohydrases, specifically amylase, produced in the salivary glands, pancreas, and small intestine. Amylase breaks down complex carbohydrates like starch into simple sugars like glucose. Second, proteases. Produced in the stomach, pancreas, and small intestine, these slice up large proteins into individual amino acids. And finally, lipases. Produced in the pancreas and small intestine, they break down lipids, which are your fats and oils, into fatty acids and glycerol. Commit these big 3 to memory, and you've got a massive chunk of your revision totally sorted.
Section 3, the vital role of bile, and spoiler alert, it is definitely not an enzyme.
Looking back at those accessory organs, the liver and gallbladder are responsible for bile. Now, listen up, because this is a classic exam trap. Bile is a vital substance for digestion, for sure, but it is definitively not an enzyme. It doesn't chemically break bonds. So, what does it actually do? Well, bile has 2 main roles. First, it's strongly alkaline. When highly acidic food leaves the stomach, bile swoops in and neutralizes that hydrochloric acid, creating the absolute optimum pH 8 environment for the enzymes in the small intestine to do their thing. Second, it emulsifies fats. Imagine a giant blob of oil. Bile breaks that large fat droplet into tiny little droplets. It's kind of like tearing a huge sheet of paper into confetti. It drastically increases the overall surface area, allowing the lipase enzymes to chemically break down the fat much, much faster.
Let's move to section 4 and see how this builds into the tubing model practical experiment.
All right, we've covered the theory, so let's look at the required practical experiments examiners love to test. It's actually incredible how perfectly this simple lab setup mimics the human body. Imagine placing a mixture of starch and an enzyme inside some partially permeable tubing, and then suspending that in a test tube of water, kept right at 37 degrees Celsius, body temperature.
Now, what's really interesting about this is what the parts represent. In an exam, you'll need to know that the partially permeable tubing represents the small intestine, and the surrounding water represents your blood plasma. And the results of this model? They perfectly prove our entire concept of digestion. If you just put starch in the tubing on its own, it stays there. The molecules are just too large to escape. But when you mix it with the amylase enzyme, the enzyme breaks the starch down to form simple sugars. Suddenly, those sugar molecules are small enough to pass right through the microscopic holes in the tubing and into the water. And we know this for sure because if we test the outside water with Benedict solution, which tests for sugar, it'll turn a positive brick red. Chemical digestion successfully solves the physical problem of absorption. Boom.
Section 5. Intestine adaptations, or how we maximize absorption. Okay, so the enzymes have done their job and the food is now broken down into tiny, soluble molecules. But how do we get them into the bloodstream as quickly and efficiently as possible? Well, the inside of the small intestine isn't just a smooth pipe. It's completely covered in millions of tiny, finger-like projections called villi. These villi are absolute structural masterpieces of biology. First off, they provide a massively large surface area, which is increased even further by microscopic microvilli on their surface. Second, the wall of the villus is literally just one single cell thick, providing an incredibly short diffusion distance for the molecules to travel. And third, they have a really rich network of blood capillaries to maintain a steep concentration gradient, rapidly whisking away the glucose and amino acids. Interestingly, the fatty acids and glycerol are actually absorbed into a specialized lymph vessel right in the center of the villus called a lacteal. And just like that, the journey from large, insoluble bites of food to tiny, soluble molecules in your blood is complete. But I want to leave you with this final, bigger picture thought. Now that you've digested and absorbed all these brilliant little molecules, how will your individual cells actually use them to keep you alive? Well, those simple sugars are going to be rushed straight to your cells to fuel aerobic respiration, giving you energy. And those amino acids, they'll be reassembled to build brand new proteins, antibodies, and even new enzymes. You are, quite literally, building your body out of the broken down bits of your lunch. Fascinating, isn't it? Keep that bigger picture in mind as you revise, and you're absolutely going to crush your GCSE exams. See you next time.